CELLULAR Evolution Chapter 3. Chapter 3. Origin of Life and Cellular Evolution. Life originated 3.8 billion years ago in Earth's history, and those first organisms were ancestors of present-day organisms which we see today. As per current understanding, the chronological order events in history of life can be represented as 1. Synthesis of organic molecules. 2. Origin of self-replicating molecules such as RNA. 3. Natural selection of suitable RNA/DNA. 4. Evolution of first prokaryotic cell. 5. Evolution of first eukaryotic cell. And 6. Evolution of multicellular eukaryotes. The organisms most familiar to us are macroscopic and multicellular, mainly plants and animals. However, for the first 3/4 of evolutionary history, Earth's only organisms were microscopic and unicellular. Earth was formed around 4.5 billion years ago, and life probably began only a few hundred million years later. There is no evidence of life for 0.7 billion years after origin of Earth. This was explained by the fact that after origin of Earth, rocks present in Earth gets melt and after cooling gets arranged according to their density, a process which was called differentiation. Scientists have found isotopes of carbon that seems to represent the metabolic activity of organisms in 3.8 billion year old rocks in Greenland. One would guess from the relatively simple structure of the prokaryotic cell that the earliest organisms were prokaryotes, and fossil records also support this presumption. Evidence of ancient prokaryotic life has been found in rocks called stromatolites. Stromatolites are banded domes of sedimentary rock that are similar to layered mats. Figure 13. Figure 13. Fossilized mats known as stromatolites. The bands seen in this section of a mat are layers of sediment that adhere to the sticky prokaryotes, which produce the succession of layers by migrating upwards. Fossils resembling spherical and filamentous prokaryotes have been found in 3.5 billion year old stromatolites in southern Africa and Western Australia. Figure 14. These are currently the oldest known fossils of living organisms. On the basis of these fossil evidences, it is considered that life on Earth originates between 3.5 to 4.0 billion years ago. Figure 14. This fossilized filamentous prokaryote, about 3.5 billion years old, was found in a stromatolite collected in Western Australia. 5.1. Major events in the history of life. Before we focus on the question of how life on Earth began, let's preview some major episodes of biological history in context of geological time. Figure 15. Fossil evidences as well as laboratory researches on the kind of chemical reactions that may have taken place on early Earth to give rise to molecular building blocks of life, such as amino acids, sugars, and nucleotides, suggest that first life form, which was prokaryotes, originated within a few hundred million years after Earth's crust cooled and solidified, beginning a succession of microorganisms that were the only forms of life on Earth for about 2 billion years. These microbes were anaerobic because there was no oxygen in air. RNA sequence data show that two distinct groups of prokaryotes, bacteria and archaea, diverged early in the history of life. On the basis of fossil records and molecular data, this split occurs between 3.0 to 2.0 billion years ago. Oxygen revolution, which was production of free oxygen in atmosphere by early photosynthetic prokaryotes, takes place about 2.5 billion years ago. It was turning point during the history of life, and it has laid the platform of evolution of aerobic life. As prokaryotic evolution continued, some organisms were able to tolerate the corrosive effects of atmospheric oxygen and became the first to use oxygen to metabolize organic molecules, and it was found that prokaryotes are more metabolically diverse as compared to eukaryotes. The oldest fossils that are definitely eukaryotic are about 1.7 billion years old and were found in an abandoned mine in Michigan. The fossils are corkscrew-shaped and closely resemble simple algae. The main criterion for distinguishing eukaryotic cells from prokaryotic cells in fossils is cell size. Complex eukaryotic cells may have evolved from simpler prokaryotic cells because complexity of organization increases the ability to attain large size. Eukaryotic cells are typically larger than prokaryotic cells. Figure 15. Some major episodes in the history of life. The timings of some events are based on fossil evidences and some are based on molecular clocks. Molecular clock studies suggest that eukaryotes originated in between 2.2 to 1.7 billion years range. Evidences also support endosymbiotic theory of evolution of eukaryotic cells from symbiotic community of prokaryotes, which has created membranous organelles, mitochondria, and chloroplasts from small prokaryotic cells living within larger prokaryotic cells. These primitive eukaryotic cells was unicellular, and the name protist is given to the group of unicellular eukaryotes, and some closely related multicellular organisms. Multicellular life is used to refer not simply to the presence of more than one cell in an organism, but to more than one kind of cell, i.e., cell differentiation. Multicellular organisms develop from a single-celled zygote to an adult with specialized cell types. The origin of development is an important step in evolution of life. All life forms with cell differentiation are eukaryotic. The molecular clock suggests that multicellular life originated about 1.5 billion years ago. However, the oldest known fossils of multicellular eukaryotes are of relatively small algae that lived about 1.2 billion years ago. Figure 16. Larger organisms such as jellyfishes and worms does not appear in the fossil records until several hundred million years later, in the late Precambrian era about 600 million years ago. Chinese paleontologists recently described a particularly rich fossil 570 million years old, containing a diversity of algae and animals. Figure 16. Fossilized alga about 1.2 billion years old. A snowball Earth hypothesis was proposed which explains that glaciers covered the planet's land masses from pole to pole. This hypothesis was supported by geologists which had recently reported evidence of a severe ice age from 750 to 570 million years ago, and it helps to explain why multicellular eukaryotes were limited in diversity and distribution until the very late Precambrian. During this period seas also would have been ice-covered. Thus, most life would have been confined to areas near deep-sea vents and hot springs. According to this hypothesis, the fossil record for the first major diversification of multicellular eukaryotes corresponds to the time of end of snowball Earth. A second radiation of eukaryotic forms produced most of the major groups of animals during the early Cambrian period, the first period of Paleozoic era. However, Cnidarians and Porphyrins were already present in late Precambrian, but a much greater diversity of animals evolved just after the start of Paleozoic era 543 million years ago. Figure 17. The colonization of land was an important milestone in the history of life. Fossil evidences suggest that terrestrial life existed only in microbial form, cyanobacteria and photosynthetic prokaryotes, over a billion years ago until the land was colonized by plants and fungi. However, macroscopic life in the form of plants, fungi, and animals did not colonize land until about 500 million years ago. Plants colonized land in company of fungi. Even today evidences are there showing association of fungi with roots of most plants. Such symbiotic association of plants and fungi are evident in some of the oldest fossilized roots. Land plants are more closely related to a group of green algae called Carophysians. Figure 18. Figure 17. The Cambrian radiation of animals. Early fossil spores of 475 million years ago have a tetrad structure that is found in some modern bryophytes, and most early plant fossils seem to be related to the phylogenetic branches leading to modern bryophytes and pteridophytes. The main events in the early evolution of land plants were the evolution of a resistant spore stage, then the evolution of vascular tissue, followed by roots and then leaves. Figure 18. Phylogeny of the main groups of plants. Plants transformed the landscape, creating new opportunities for all life, including herbivorous animals and their predators. Although many animal groups are represented in terrestrial environments, the most widespread and diverse land animals are certain arthropods and certain vertebrates. The terrestrial vertebrates are called tetrapod, a reference to the 4 walking limbs that distinguish them from the various aquatic vertebrates. An excellent fossil record documents the descent of amphibians from fishes. Reptiles evolved from amphibians and both birds and mammals evolved from reptiles. 3.2. The origin of life, biopoiesis. The question of how life began is more specifically about the genesis of prokaryotes. Sometimes between about 4.0 to 3.5 billion years ago, when the planet was inhabited by bacteria advanced enough to build stromatolites, the first organisms came into existence. What was their origin? To explain this, many theories were proposed during different time periods, of which naturalistic or chemosynthetic origin given by A. 1. Oparin and J.B.S. Haldane in 1920 was widely accepted. Before looking to Oparin and Haldane theory, let us discuss some of the other theories in brief. 3.2.1 Theory of Special Creation. It states that life was created by some supernatural power. This theory was based on religious thoughts and was proposed by Father Sorge and supported by Milton. It is now reputed by most of evolutionary biologists as it has no room for change and adaptation. It assumes species to be static. 3.2.2 Theory of Spontaneous Generation, Abiogenesis. From the time of the ancient Greeks until well into the 19th century, it was common knowledge that life could arise from non-living matter. This idea of life emerging from lifeless material is called spontaneous generation. This theory was supported by Greek philosophers like Anaximander, 611 to 547 BC, Aristotle, 384 to 322 BC, Thales, 624 to 547 BC, Xenophanes, Plato, 427 to 347 BC, etc. They believe that snails, fishes, frogs-like organisms are spontaneously from mud. It was believed that not only life originated spontaneously in past but that it is originating even today. Cases were cited of the production of grasshoppers, many other insects, and even mice and toads from a rainy spelly. The 17th century abiogenesis theory was supported by von Helmont, 1642, who claimed that mice formed in 21 days from a dirty, sweat-soaked shirt in a wheat barn in the dark. 32.3 Theory of Biogenesis. According to theory of biogenesis, life originates from pre-existing life. This theory was supported by some scientists like Francisco Redi, 1668, Lazzaro Spallanzani, 1767, Louis Pasteur, 1862, Harvey, 1651, and Huxley, 1870. Louis Pasteur's famous experiments with broth completely overturned the concept of spontaneous generation, even for microorganisms. As far as we know, all life today arises only by the reproduction of pre-existing life. This life from life principle is biogenesis. But what about the first organisms? If they arose by biogenesis, then they couldn't have been the first organisms. So this put a question mark on the theory of biogenesis. To solve this puzzle, A.I. Oparin and J.B.S. Haldane put forward their chemosynthetic theory of origin of life. 3.2.4 Primitive environment and chemical evolution. Most biologists support the hypothesis that life on Earth developed from non-living materials, abiogenesis, that became ordered into molecular aggregates that was eventually capable of self-replication and metabolism. As far as we know, life cannot arise by spontaneous generation from inanimate material, today, but conditions were very different when Earth was only a billion-year-old. Primitive Earth's atmosphere was totally different from present-day atmosphere. Molecular free atmospheric oxygen gas, O2, was absent, but H2, NH3 and H2O vapors were present, due to which atmosphere was reducing. Lightning, volcanic activity, meteorite bombardment, and ultraviolet radiation were all more intense than what we experience today. In that ancient environment, origin of life was possible. It was considered that first organisms were product of a chemical evolution in 4 stages. 1. The abiotic synthesis and accumulation of small organic molecules, or monomers, such as amino acids and nucleotides. 2. The joining of these monomers into polymers, including proteins and nucleic acids. 3. The aggregation of abiotically produced molecules into droplets, called protobionts, that had chemical characteristic different from their surroundings. 4. The origin of heredity and multiplication. 6.3 Operin and Haldane theory, naturalistic theory of origin of life. In 1920 A.I. Operin of Russia and J. B. S. Haldane of Great Britain independently postulated that condition on the primitive Earth favored chemical reactions that synthesized organic compounds from inorganic precursors present in the early atmosphere and seas. This cannot happen in the modern world, Operin and Haldane reasoned, because the present atmosphere is rich in oxygen produced by photosynthetic life. The oxidizing atmosphere of today is not conducive to the spontaneous synthesis of complex molecules because the oxygen attacks chemical bonds, extracting electrons. Before oxygen-producing photosynthesis, Earth had a much less oxidizing atmosphere, derived mainly from volcanic vapors. Such a reducing, electron-adding, atmosphere would have enhanced the joining of simple molecules to form more complex ones. Even with a reducing atmosphere, making organic molecules would require considerable energy, which was probably provided by lightning and the intense UV radiation that penetrated the primitive atmosphere. The modern atmosphere has a layer of ozone produced from oxygen, and this ozone shield screens out most UV radiation. There is also evidence that young sun emit more UV radiation than old Sunday. According to Operin and Haldane, chemical conditions and energy resources was perfect for abiotic synthesis of organic molecules. Before discussing chemical evolution, we had a look on chemical and structural changes in primitive Earth, which builds a platform for chemical evolution on Earth. 3.3.1 Origin of Earth. The Earth was formed just like other planets. In the beginning, Earth was a huge collection of cold objects called planetesimals. These planetesimals which formed the Earth came from two regions, from the regions occupied by the inner planets like Mercury and Venus, and from the farther regions of Jupiter, Saturn and Uranus. The planetesimals coming from the inner planets region consisted mostly of the compounds of iron, silicon and magnesium with traces of other elements, whereas those coming from the regions of outer planets largely consisted of matter like hydrogen, water, carbon dioxide and methane. As more and more planetesimals collided with the Earth and got stuck into it, their kinetic energy was converted into heat energy. In addition to this, the radioactive disintegration of the elements like uranium, thorium and potassium produced a lot of heat energy. The enormous heat energy thus produced, along with gravitational compression, heated the Earth so much that it melted to form a liquid. This melting of Earth happened about 800 million years after its formation. It was because of the melting of Earth that the surface records of the Earth for the first 800 million years of its existence were completely destroyed. When the Earth melted, the molten material present in it began to reorganize itself under the influence of gravity. The molten iron, being the heaviest material, fell to the center of the Earth and formed the core of the Earth, innermost part of the Earth. The lighter constituents which were earlier present in the core came up and formed the crust of Earth. The medium density matter remained in between the core and crust to form the mantle of Earth. This organization of molten Earth into various layers having different densities is called differentiation. As a result of differentiation, the Earth was divided into three major layers: crust, mantle and core. Figure 19. Figure 19, Structure of Earth. 3.3.2 Primitive atmosphere of Earth. During this differentiation process, the lightest materials present in the original Earth were expelled as gases to form the first atmosphere. The newly formed atmosphere at this stage consists mainly of gases like nitrogen, carbon dioxide, hydrogen, and water vapors. Molecular oxygen, gas, was absent in the initial atmosphere of Earth. Environment was strongly reducing. The crust is made up of light rocks, rich in silica and aluminum, The mantle of Earth consists of rocks which are largely made up of iron silicate and magnesium silicate. The core of Earth is made up of mainly iron and some nickel. The core of Earth actually consists of two parts, the inner core and outer core. The inner core is a solid sphere of highly compressed iron, whereas the outer core is of molten iron. The temperature at the center of the Earth is about 4,000 degrees Celsius, and the pressure is extremely high, being about 3.7 million atmospheres. It is because of this extremely high pressure that the iron and inner core can remain in solid state despite the very high temperature of 4,000 degrees Celsius, which is much above the melting point of iron, 1,539 degrees Celsius. Since the pressure is comparatively lower in the outer core, therefore, the iron metal remains in the molten form in the outer part of the core of Earth. Thus, the outer core behaves like a liquid. Since the core of Earth is very hot but the surface of Earth is colder, so heat flows outwards from the hot interior of the Earth to the surface of Earth. This flow of heat generates convectional currents in the interior of Earth. These convectional currents are responsible for drifting of plates in crust region, continental drift, and thus events like volcanoes, earthquakes, formation of mountains and plateaus. After evolution of atmosphere, it was turn of evolution of water bodies, oceans. The primitive crust of Earth was constantly punctured by meteorites falling from the outer space. Now, since the atmosphere was very thin at that time, it could not slow down the falling meteorites by offering sufficient air resistance. Due to this, the meteorites fell on Earth with great force and made many holes in the newly formed crust. These holes in the Earth's surface were called vents. Through these vents, the molten rocks from upper core and lower mantle came above the surface of Earth. This molten rock, called magma, cooled down, solidified, and became a part of the crust. This process of the magma rising to the surface of Earth through vents releases more gases to the atmosphere. These gases were mainly hydrogen, nitrogen, carbon dioxide, and water vapors. In due course of time, a lot of water vapors was concentrated in the atmosphere. When this water vapors present in the atmosphere rose up, it got cooled. The cooled water vapor turned into clouds and then poured on the Earth in the form of rains. This rainwater collected in all low-lying areas of the Earth crust and formed oceans. Formation of oceans was key point in origin of first life form because life originates in sea water. 3.3.3 Synthesis of simple organic compounds. Initially, gases like hydrogen, carbon dioxide, and nitrogen were abundant gases in primitive Earth. These gases interact with each other to form methane, ammonia, hydrogen cyanide, nitrides, carbides, and various other gases in dissolved form in water. Methane was probably first organic compound and hydrogen cyanide was formed later. 3.3.4 Synthesis of simple organic monomers. High concentration of these organic compounds in seawater induced them to react with each other and produce some unsaturated hydrocarbons like ethylene, aldehydes, ketones, alcohols, and organic acids. As a result of further interaction of these saturated and unsaturated compounds, organic monomers like simple sugars, glucose, ribose, amino acids, fatty acids, glycerol, and nitrogenous bases were synthesized. CH4 plus NH3 plus H2O amino acids. CH4 plus HCN plus NH3 plus H2O purines plus pyrimidines. CH4 plus H2O sugar plus glycerol plus fatty acids. Haldane described the sea-containing molecules of these organic substances in abundance as the hot dilute soup or primitive broth that has been nicknamed Haldane soup. In 1953, the first support to operin and Haldane theory came from Stanley Miller and Harold Urey. They created primitive Earth's environmental condition in laboratory and built up an apparatus of glass tubes and flasks as shown in Figure 20. Figure 20, Miller and Urey experimental apparatus. He created the atmosphere consisted of water, hydrogen, methane, and ammonia. Sparks were discharged in the synthetic atmosphere to mimic lightning. A condenser cooled the atmosphere, raining water and dissolved compounds back to the miniature sea. As material circulated through the apparatus, the solution in the flask changed from clear to murky brown. After 1 week, when the contents of the solution was analyzed, a variety of organic compounds, including urea, organic acids, sugars, nitrogenous bases and amino acids, glycine, alanine and aspartate, were synthesized. After abiotic synthesis of organic monomers, the next step was aggregation of these monomers to form polymers, polypeptides, polysaccharides, nucleic acids. 3.3.5 Synthesis of complex organic polymers. Today's synthesis of these polymers takes place inside the cell, and it is considered as an anabolic process which requires energy in form of ADP, and such reactions are catalyzed by enzymes, but in primitive environmental conditions abiotic synthesis of these polymers would have had to occur without the help of these enzymes. Moreover, the dilute concentration of these monomers in seawater would not favor spontaneous polymerization reactions. However, in laboratory experiments, polymerization does occurs when dilute solutions of organic monomers are dripped onto hot sands, clay, or rocks. This process concentrates the monomers as water vaporizes. Sidney Fox of the University of Miami, by using such methods, synthesizes polypeptides by abiotic means. Such abiotically produced polypeptides was called proteinoids or microspheres by Sidney Fox. Perhaps waves or rain splashed dilute solutions of organic monomers onto fresh lava or other hot rocks on the early Earth, and then rinsed proteinoids and other polymers back into the water. It was also found that clay have been especially important as a substratum for the polymerization reactions, a prerequisite to life. Clay particle posses charged sites which assist amino acids and other monomers to bind with electrostatic interactions, and thus concentrates monomers from the dilute solutions. At some binding sites, metal ions, such as iron and zinc, function as catalysts, facilitating the reactions that link monomers. Even today, these metal ions play an important role in catalysis and metalloenzymes. 3.3.6 Formation of protobionts or protocells. Abiotically synthesized polymers aggregates to form protobionts. These are considered to be the first life form, and living cells may have been preceded protobionts. They exhibit some properties of life, like they are capable of precise reproduction and can maintain their internal environment different from their surrounding, as well as metabolism and excitability. One type of protobiont is coacervate, named by operin, is a stable droplet that tends to self-assemble when a suspension of macromolecules, polypeptides, nucleic acids, and polysaccharides is shaken. Figure 21a. Coacervates are surrounded and stabilized by a shell of water molecules. If enzymes are incorporated in coacervates, then they are able to absorb substrates from their surroundings and release the products of the reaction catalyzed by the enzymes. It is believed that coacervates can divide if they attain large size. Figure 21b. Another type of protobiont is microsphere. Figure 21c, named by Sidney Fox, are formed when water is added to thermal proteinoids. They are coated by a protein membrane which is selectively permeable. Figure 21a, coacervate B, division in coacervates, and C, microsphere. Microspheres undergo osmotic swelling or shrinkage when placed in solutions of different salt concentrations. Some microspheres also store energy in form of membrane potential. They can also discharge potential in Hervé-like fashion. Such excitability is characteristic of all life. 3.3.7 Formation of first cell and biological evolution. The nucleic acids in pre-cells had the capacity to multiply, but gradually it started directing a series of chemical reactions like protein synthesis. That was the significant step in the transformation of pre-cell into a cell. This chemical evolution gave rise to the biological evolution. The first cells or primitive cells were marine and anaerobic, as molecular oxygen was absent. They were chemoheterotroph, saprotroph, in nature. Due to continued withdrawal of organic molecules by chemoheterotrophs, organic material decreased in oceans. Before the organic Material disappeared and c. The new modes of nutrition developed. One of them was chemosynthesis. The organism which perform chemosynthesis are called as chemoautotrophs. They were anaerobic and synthesized organic molecules from inorganic material. The energy was obtained by oxidizing inorganic materials present in the sea. This event helped in the transformation of heterotrophs into autotrophs. Such mode of nutrition is found in bacteria, e.g. sulfur bacteria, nitrifying bacteria. But very soon all inorganic material in sea will also deplete, so it was the favorable circumstances for mutation, which resulted in the development of chromophores/bacterial chlorophyll. The chromophores had the ability to trap light energy and convert it into chemical energy. Newly evolved organism could absorb solar energy and convert it into chemical form. These organisms called photoautotrophs. They were anaerobic and utilized hydrogen from sources other than water like H2S. Therefore, no oxygen was evolved and atmosphere remained. Reducing. This stage of photosynthetic autotrophism is represented by planktonic sulfur bacteria of today. The bacterial chlorophyll by molecular changes formed true chlorophyll. Such organisms which were bearing true chlorophyll are similar to today's cyanobacteria, blue-green algae. They utilized water as hydrogen donor and evolved oxygen. Liberation of free O2 by blue-green algae like prokaryotes due to photosynthesis was a revolutionary change in the history of Earth. It is called oxygen revolution. It includes important changes like 1. Atmosphere of Earth changed from reducing to oxidizing, hence possibilities of further chemical evolution and abiogenesis ends, because chemical evolution always take place in reducing environment. 2. Free as oxidized CH4 and NH3 to form gases like CO2, N2 and H2O. 3. Accumulation of FeO2 formed a layer of O3, ozone, above the atmosphere of Earth, which started absorbing most of the UV rays of sunlight. 3.4 First genetic material RNA world. After synthesis of first life form i.e. protobionts, the major thing was perpetuation of these protobionts. These droplets uptake nutrients in the form of abiotically synthesized monomers from the surrounding environment, seawater, and divides to give their progeny. As these droplets grew, and split again, their unique catalysts and other functional biomolecules would increasingly diluted. Natural selection favors droplets which develops the mechanism to copy their biomolecules, i.e. mechanism of heredity, and then distribute equally before splitting to give the progeny cells. Since we know that metabolism is a key feature of life and all metabolic processes are governed by enzymes present inside the cell, the information regarding synthesis of enzymes lies in genetic material which is DNA in the modern cells. It was experimentally proved that even protobionts possess metabolic activity. It means that protobionts have some biomolecules having catalytic activity. If these biomolecules were enzymes, then there must be DNA present to encode them. Khorana, 1970, artificially synthesized 77 nucleotide RNA molecule outside a living cell, which suggests that probably RNA was the primordial genetic material rather than DNA. Zog, Thomas Cech and Altman described that some RNA molecules have enzymatic activity. Hence probably the RNA enzymes called ribozymes were able to replicate the primordial RNA. The discovery of RNA molecule working as enzyme has also changed our thinking about origin of life. It is now believed that about 4 billion years ago Earth was an RNA world in which RNA molecule carried out all the process of life without the help of either protein or DNA. Now it is assumed that there is no need to have complex cell-like structure to be designated as life form. In primitive time, RNA acted both as genetic material as well as an enzyme and was capable of self-replication. Thus, this naked RNA, like virids, can represent the first life form. Due to intramolecular hydrogen bonding between nucleotides and RNA, it can form various types of 3D structures, and each type of structure has a unique conformation resembling proteins and enzymes and can catalyze a metabolic reaction. The RNA molecule thus has both a genotype, its nucleotide sequence, and a phenotype, its conformation, which interacts with the surrounding molecules in a specific ways. Later on, RNA started to act as template on which amino acids can arrange and facilitate synthesis of peptide bonds to produce polypeptides. If RNA happened to synthesize a polypeptide that in turn behaved as an enzyme helping the RNA molecule to replicate, then the early chemical dynamics included molecular cooperation as well as competition. Figure 22. Hypothesis for the beginning of molecular cooperation. This first step toward the replication and translation of genetic information may have been taken by molecular evolution even before RNA and polypeptides became packaged within membranes. Figure 22a. The packaging of primitive RNA and their polypeptide products within a membrane would have been a significant milestone in the early history of life, and polypeptides facilitate replication of RNA enclosed in limiting membrane rather than competing RNA molecules in its environment, i.e. external to limiting membrane. Figure 22b. It is well understood that RNA is chemically less stable than DNA because it is rapidly hydrolyzed even if there is slight pH fluctuation. Therefore, RNA cannot be considered as a stable genetic material. Meanwhile, in some primitive cells, ribose sugar give rise to deoxyribose sugar, and uracil give rise to thymine, and this change inside the cell is responsible for synthesis of DNA. DNA is chemically more stable than RNA, due to presence of 2'-deoxyribose sugar which lacks free 2-OH group. Thus, DNA started to act as genetic material and RNA started to act as intermediate in transferring the message given by DNA for synthesis of proteins and enzymes. Cells which had DNA as genetic material were more stable as compared to cells having RNA as genetic material, and thus, they outcompeted RNA-based cells by utilizing more resources from surrounding DNA-based cells were positively selected by natural selection, and gradually only those cells survived which were having DNA as a genetic material. 2.5 Evolution of DNA molecules. The RNA molecules would have been subjected to evolutionary processes that eventually gave rise to DNA molecules. In the first stage, the development of protein enzymes may have taken place to substitute the catalytic properties of ribozymes. How the transition from RNA to protein could have occurred has raised questions that cannot be answered. Possibly, the proteins became coding molecules that could specify catalytic proteins. This creates two scenarios for the evolution of the first coding RNA. A ribozyme could have evolved to become the coding molecule, so as to have a dual catalytic and coding function, or coding molecules could have been synthesized by ribozymes. The theories about the origins of translation and the genetic code suggest that ribozymes were probably synthesizing the coding molecules. DNA are more stable as genetic material as compared to RNA, thus in the next stage, we have to envisage transfer of the coding function to the DNA molecules. This would involve reduction of ribonucleotides giving deoxyribonucleotides, which could then be polymerized into copies of DNA by a reaction catalyzed by enzymes similar to reverse transcriptase. Uracil would have been replaced by its methylated derivative thymine, providing mismatch repair machinery to work in an efficient way. The DNA molecules performing coding function would have become double-stranded duplexes in response to the possible requirement for repair of DNA damage by copying the partner strand. The first DNA molecules can be visualized as specifying a single protein, each therefore representing a single gene. How the single genes became linked to form separate chromosomes is not understood, though several different mechanisms have been proposed. The evolution of living forms was accompanied by an increase in complexity of genomes. Gene number increased from about 1,000 in bacteria to 80,000 in vertebrates and humans. Instead of a gradual increase, there seem to have been two dramatic stages of expansion in gene numbers. The first expansion occurred about 1.4 billion years ago with the appearance of eukaryotes, when the few thousand genes present in prokaryotes increased to above 10,000 in the simplest eukaryotes. The second stage of expansion probably occurred when the first vertebrates appeared, with acquisition of about 50,000 genes. 3.6. From microorganism to man. The first cells, which were similar to present-day bacteria, are believed to have evolved 3.8 billion years ago. The earliest living organisms were prokaryotes, is based on the discovery of microfossils from early Precambrian rocks. The first living beings on Earth were anaerobic, because there was no oxygen in air. About 2.7 billion years ago, conditions become suitable for aerobic respiration with the release of free CO2. Aerobic respiration yields about 20 times more energy than anaerobic respiration, hence the prokaryotes adapted themselves for aerobic mode of respiration. Table 3. Geological time scales showing evolutionary changes from microbe to man. Nucleus, mitochondria, and other cell organelles. Developed in the cell and free-living eukaryotic cell-like organism originated about 1.5 billion years ago in the primitive ocean. Evidence from fossil records suggests that the first eukaryotic cells arose about 1.4 billion years ago, in the form of structures resembling single-celled algae. The first multicellular algae came into existence about 0.9 billion years ago. Around 640 million years ago, multicellular animals seem to have appeared. During the Cambrian period, about 530 million years ago, invertebrate life seems to have proliferated into many new forms. The first animals were single-celled protozoa. The worms and sponges seem then to have evolved in the seas. Then came the hard-shelled crustaceans, which left many fossils of their bony armor. The mass extinction, which occurred 500 million years ago, wiped out most of the new forms. Evolution thereafter is seen to have established insects, animals, and plants by 350 million years ago. The first land plants, Coelopsids and Lycopsids, appeared in the Silurian of Paleozoic, followed by forests of gymnosperms, seed ferns, and bryophytes. The earliest amphibians arose in the age of the forests. The flowering plants came later in the Mesozoic, Table 3. The earliest vegetables vertebrates appeared in early Paleozoic about 300 million years ago. The fishes, amphibians, and reptiles also arose, in that order, during the Paleozoic. The reptiles ruled the earth for many millions of years. During the Mesozoic, giant-like reptiles, the dinosaurs, were dominating the earth for over 1 million years, then became extinct. From the reptiles, the birds and mammals seem to have diverged along two separate lines, the mammals culminating in man. The lowermost animals with backbones on the evolutionary scale belong to the chordates, possessing either a notochord or a vertebral column. The first chordates are not preserved as fossils as they lacked a bony armor. The present-day sea lancelet Amphioxus is perhaps a living representative of the primitive chordates. It has a notochord, a nerve cord, and a digestive tube. There is no head, brain, or sense organ, except pigment spots sensitive to light. The earliest vertebrates were jawless, of which the living form lamprey is a good example. The fossil record shows how amphibians and reptiles evolved. The first mammals seem to have descended from mammal-like reptiles in the Jurassic. These four-footed mammals with uniform body temperature and high basic metabolism are also referred to as warm-blooded animals. The last to come in evolution of vertebrates are the primates, including the lemurs, tarsiers, monkeys, apes, and man. The more advanced primates are comprised of monkeys and apes. The apes have no external tail and have evolved as brake-eating animals, swinging on trees by their arms. This type of locomotion is predominant among the apes. The modern apes include the gibbons, orangutan, of East Indies, and the chimpanzees and the gorillas, the largest in size, of Africa. The earliest organism resembling human beings was Australopithecus, an erect, biped walking, tool-making creature discovered from cave deposits of Pleistocene in S. Africa more than 10 million years ago. It was followed by Homo habilis, 9 million years ago, Homo erectus, 5 million years ago, Homo sapiens neanderthalensis, 2.3 million years ago, and finally Homo sapiens sapiens, modern man, which appeared about 10,000 years ago in Central Asia. 3.7. Controversy about the origin of life. While most researchers agree that life first appeared as the primitive, but there is little agreement as to just where this occurred. During early Earth from 4.6 to 4.0 billion years ago, the surface was molten hot, and no life is expected. Gradually temperatures dropped, and by about 3.8 billion years ago, ocean temperatures are thought to have dropped to a hot 49 degrees to 88 degrees Celsius. Between 4.0 and 3.8 billion years ago, life first appeared, promptly after the Earth was inhabitable. It is believed that early atmosphere contained principally carbon dioxide, CO2, and nitrogen gas, N2, along with significant amounts of water vapor, H2O. It also contained hydrogen gas, H2, and compounds in which hydrogen atoms were bonded to the other light elements, sulfur, nitrogen, and carbon, producing hydrogen sulfide, H2S, ammonia, NH3, and methane, CH4. We refer to such an atmosphere as a reducing atmosphere because of the ample availability of hydrogen atoms and their electrons. In such a reducing atmosphere, it would not take as much energy as it would today to form the carbon-rich molecules from which life evolved. The key to this reducing atmosphere hypothesis is the assumption that there was very little oxygen around. Critics of the reducing atmosphere hypothesis point out that 1.NO carbonates have been found in rocks dating back to the early Earth. This suggests that at that time carbon dioxide was locked up in the atmosphere, and if that was the case, then the prebiotic atmosphere would not have been reducing. 2. If a prebiotic reducing atmosphere would have been oxygen-free, there would have been no ozone. Without the protective ozone layer, any organic compounds that might have formed would have been broken down quickly by ultraviolet radiation. The origin of life remains a matter of scientific speculation, and there are alternative views of how several key processes occurred. 1. Deep in the Earth's crust. One hypothesis suggests that life originated deep in the Earth's crust. In 1988, Gunter Wachtschauser proposed that life might have formed as a byproduct of volcanic activity, with iron and nickel sulfide minerals acting as chemical catalysts to recombine gases spewing from eruptions into the building blocks of life. 2. Within clay, other researchers have proposed the unusual hypothesis that life is the result of silicate surface chemistry. The surface of clays have positive charges to attract organic molecules and exclude water, providing a potential catalytic surface on which life's early chemistry might have occurred. 3. At deep-sea hydrothermal vents, recent studies on archaea suggest that life could have originated in deep hydrothermal vents. This hypothesis is becoming more popular, since the necessary prebiotic molecules being synthesized on metal sulfides in the vents. The positive charge of the sulfides would have acted as a magnet for negatively charged organic molecules. In part, the current popularity of this hypothesis comes from the new science of genomics, which suggests that the ancestors of today's prokaryotes are most closely related to the bacteria that live on the deep-sea vents. 4. Theory of panspermia. It is possible that some organic compounds reached the early Earth from space. This idea, called panspermia, explains that hundreds of thousands of meteorites and comets hitting the early Earth brought with them organic molecules formed abiotically in outer space. Extraterrestrial organic compounds, including amino acids, have been found in modern meteorites, and it seems that these bodies could have enriched the early Earth with organic compounds. Both panspermia and chemical evolution could have contributed to the pool of organic molecules that formed the earliest life, but many scientists believe that extraterrestrial sources made only a minor contribution.